So in this project, we've designed new base pairs for DNA with a new structure altogether. Several people in the literature over the last 5 to 10 years have started to design new base pairs for DNA to try to expand the genetic code. Our goal is a little bit longer term than that, and that is to come up with an entirely new architecture for DNA. And so we call this architecture expanded DNA, where we actually increase the size of DNA bases and base pairs. By doing so, we actually stretch out the individual bases using chemistry. So we use synthetic chemistry to design and synthesize DNA bases that are larger than normal ones. And so base pairs are larger than normal ones. In order to do this, we designed 4 new bases, and these pair with the 4 natural ones. So we end up with 8 letters of genetic information, and so it's much more complex than normal DNA. So this expanded DNA is something we came up with back in about 1999 or 2000 was the idea. And we started working on it in around 2000 and made our first helices of expanded DNA in around 2003 or so. So we were happy to find out that you actually can form double helices based on expanded base pairs. That the DNA was formed a helix, just like normal DNA, except it's bigger. Right. And the cylinder is larger. And not only is it formed stably, but it actually is more stable than natural DNA. So it takes more energy to unwind the double helix of xDNA than it does natural DNA. Another nice property of xDNA is that it's fluorescent, and that actually may have some bearing on applications of xDNA. So our long-term goal for this project has been to see how lifelike we can make xDNA. By that I mean what sorts of biophysical, biochemical, and ultimately biological properties this molecule can have. Can it carry information the same way DNA does in a sequence? Can this sequence be transmitted to a daughter sequence? That's called replication. And so we want to try to be able to replicate xDNA. Ultimately, we'd like to be able to evolve xDNA the same way that you can DNA now in a test tube.
And in terms of genetic engineering, do you plan on using the xDNA to create novel proteins?
Yeah, that's a really hard problem. Our first goal is simply to see if we can get xDNA to make copies of new xDNAs inside a cell. And that's about a 5 to 10 year problem, I think. Right. We have to think long term when it's big, tough problems like this. So the problem with making proteins is you have to engineer lots of parts of the cells in order to make that happen, because you would have to make RNA copies of xDNA and turn it into xRNA. You would have to then have an expanded ribosome, this machine that copies RNA and makes it into proteins. And that's a really hard problem. The ribosome is an extensively complicated machine. It's huge. And so engineering that one is a little harder, but not impossible. Actually, there are people that are working on methods for engineering ribosomes. So that could be done ultimately. I'll be happy if we can simply make xDNA into an evolving system. And so that's one of our goals. You mentioned the possibility of applications. Mm-hmm. And so while I'm very interested in just the scientific question of whether DNA in other forms can exist and function, that's more of a blue sky question. We're also interested in potential applications. So the fact that xDNA is fluorescent and changes its fluorescence when it binds to DNA or RNA suggests its use to diagnose sequences, to find genetic sequences in cells and give some signal when that happens. Mm-hmm. So that's something we'd be interested in exploring. The fact that it binds more tightly than natural DNA or RNA also makes it potentially very useful as a way of pulling out genetic sequences very efficiently from big biological mixtures. So there are some properties of xDNA that make it, I think, potentially interesting for applications also. There's a really interesting, so some theoreticians have gotten hold of xDNA in their computers, and they're doing calculations on what they predict the properties of xDNA will be. And they predict that it actually may be a wire, that it may be a single, so a single helix of xDNA may be able to conduct charge much better than DNA can. DNA is a terrible conductor of electricity. But because of the larger structure of xDNA and its base pairs, they predict that actually will be able to carry charge much better. And so that's potentially really interesting too. You could imagine making circuits out of xDNA that just form themselves, right? Because DNA can form helices in a programmed way.
Right.
So if one could do that and make circuits that form themselves, I think that would really be exciting.